Welcome to the Organic Chemistry Podcast, Dr. Brian Lloyd's scribblecast of organic chemistry lectures and solutions to homework problems. In this scribblecast lecture, we'll be examining peptides and proteins. A peptide consists of two or more amino acids covalently attached by what is called the peptide bond. Now, the peptide bond is simply an amide functional group. The amide functional group links an alpha amino group of one amino acid with the alpha carboxyl group of another amino acid. Each amino acid group in the peptide is referred to as an amino acid residue. Now, the amino acid with the free NH3 minus group, so the end of the peptide that has the free NH3 minus group, is referred to as the N-terminal amino acid or N-terminal residue. The peptide end, the amino acid in the peptide that has the free CO2 minus group, is referred to as the C-terminal residue or the C-terminal amino acid. Now, let's consider two amino acids that come together and react in this way. Let's start with the Zwitter ion, say, of alanine. And let's react this with the Zwitter ion of methionine. So we have CH2 and we have two of those. Then we have an S and a CH3. What do we get? Well, the N-terminal residue is the alanine. The CO2 minus links up with the NH3 plus to make an amide bond. The C-terminal amino acid residue is the methionine. So we have the CH3 for the alanine, and we have the two CH2s, S and the CH3 of the methionine. Our amide functional group is right here, and you can see how the N of one amino acid linked up with the CO2 minus of the other. Alanine has the N-terminal amino acid residue, while methionine has the CO2 minus, which is a C-terminal amino acid residue. Now, the C-terminal amino acid is given the root name. So, in fact, this molecule would be named methionine. All other substituents are named in order of appearance. As they appear to the left, the "-ine" ending is dropped and YL is added. So alanine becomes alanyl. It's very important to note that the order is important. Sometimes, rather than drawing out the structure, an abbreviated version using the abbreviations of the amino acids is shown, where ALA would be drawn with a bond to MET. Note that ALA bonded to MET is a different peptide structurally than MET bonded to ALA. So order is important. Those two are different. Okay. Now, let's talk about the actual bonding in the peptide, and then we'll talk about the types of peptides that can be found. Again, the bonding in the peptide is the amide functional group. An amide bond we've already discussed. That is, we have an RC double bond O, NH, R. There's a lone pair of nitrogen, but that lone pair is not basic. Not only is it not basic, but it has huge impact on the structure of peptides. It is resonance stabilized. The lone pair can dump in to create a pi bond. A pi bond of C double bond O moves out. And we now get an O minus. Because of charge separation, this is a minor contributor. But it does have huge implications structurally. The nitrogen's positively charged. Notice there is now a C double bond N unit in the amide. Now, neither of these resonance contributors exists. What exists is a resonance hybrid, and that means there's some double bond character between the carbon and nitrogen. Double bonds have hindered rotation, so there's some hindered rotation in this molecule that will help lock these peptides into certain orientations. In actual fact, a typical amine bond is around 1.47 angstroms in length. As expected, double bonds which are shorter. The CN bond of the amide is about 1.32 angstroms. Because of the structure, in order to reduce steric hindrance, the molecule staggers the R groups. So if this amino acid residue had the H with a wedge and the R group, let's do alanine, going into the page, then the other amino acid would put the H going into the page, and our two CH2s, SCH3 of our methionine coming out of the page. And we'd have a CO2 minus here. Now, the bulky steric chains could be referred to as being trans in orientation to each other, but remember that term is directly related to steric hindrance caused by a ring or a double bond, so it's not totally appropriate to use here, but nevertheless, the bulky groups, this R group and this R group, one is pointing into the page, one is coming out. And so they're staggered if you like. Now, peptides like carbohydrates can be broken down into several categories. The first category is oligopeptides, just like oligosaccharides. In oligosaccharides, we define them as 2 to 8 carbohydrate units. Oligopeptides are defined as 2 to 10 amino acid residues. So you're an oligopeptide if you are less than 10 amino acid residues. Okay, so for example, the molecule ceryl arginyl cysteine is an oligopeptide, and we sometimes, just like we have disaccharides, trisaccharides, monosaccharides, this would be referred to as a tripeptide. Okay. Polypeptides unlike polysaccharides, which is any sugar in excess of the oligosaccharide, greater than 10 sugars, polypeptides are limited to less than 50. So typically from 10 to about 50 residues. A special term is used for more than 50 amino acid residues, and that is the term protein. Proteins are molecules that have unique structure. They can adopt unique conformations with definite sequences and folding. These folds are caused, if you like, from the C double bond N character in the protein amide bonds. Polypeptides with greater than 50 amino acids, typically referred to as proteins. These proteins have a definite biological function. Now, peptides are formed in the gastrointestinal tract during digestion of protein by proteases. And proteases are enzymes that hydrolyze peptide bonds. Peptides are found in all types of organisms and have many functions. Now, it is very important to be able to determine peptide structure. How can one go about determining peptide structure? Well, if we want to determine peptide structure, we can actually hydrolyze peptides into individual constituent amino acids. We can do this by refluxing. Refluxing simply means boiling a solution. Refluxing at approximately 110 degrees Celsius in 6 molar HCl. For about 24 to 72 hours. If you do this, you will actually hydrolyze all of the peptide bonds. And then the amino acids can be identified by column chromatography. You can use some kind of chromatography. With thin layer chromatography, there's different types that could be used. You can also use a process referred to as electrophoresis. And electrophoresis takes advantage of the differing charge in differing amino acids at different pHs, allowing you to separate charged molecules from neutral molecules. However, we have a problem with this. This just identifies what amino acids there are. It identifies them. but it doesn't give you the order or sequence. What's the order or sequence of the amino acids? And remember, the order is very important, because if you don't know the order of the peptide, you could generate different peptides. Order is very important. So if we want to determine amino acid sequence, we have to carry out what is called terminal residue analysis. Now, in terminal residue analysis, there are two methodologies. We can analyze from the N-terminal residue. N-terminal residue analysis. Now, N-terminal residue analysis is referred to as the Edman Degradation. This is one of the processes that can be carried out that allows you to actually determine the order starting from the N-terminal residue, or the left-hand side. Now, the N-terminal residue of the peptide is reacted with phenylisothiocyanate. Let's take a look at that reaction. We have a phenyl with an N double bond C double bond S. How's that? I know it's not bent, but we'll draw it that way. And this can react with the N-terminus. Now, the zwitterion can be in equilibrium with the deprotonated form. And so you have the CH, you have your R side chain, and then you have a C double bond O, and you're linked to the rest of the peptide. Now, what can happen is the amine can attack the carbon of the thiocyanate, or the isothiocyanate, the phenylisothiocyanate. And the pi electrons can move, producing an N-, and then they can grab a proton. If I then do this in base, we will get some deprotonation. And I will produce this. And then we have our CHR, our C double bond O, and of course the rest of the peptide. Now, let's be clear about this. What we've done here is what was our phenylisothiocyanate, and the other side was our peptide. This N had originally picked up a proton, but the OH- deprotonated it. We have 1, 2, 3, 4, 5 atoms. So this lone pair could wrap around and attack, making a 5-membered ring. If we do this and lose H+ because we're in base, we will produce a 5-membered ring. Now, the atoms in the ring are thus, we've got a carbon here, And so you can tell where we are. We'll make that carbon the double at C double bond S. So this is the one with the phenyl. And then here we have our NH. And here we have our C with an H and the R. Remember the R group that designates the amino acid. And this is the carbon that was attacked or cyclized. And it has an O minus. And this C is attached to the rest of the peptide. Now what's interesting, in methyl or nitromethane, which is anhydrous as a solvent, we can cleave the attachment to the rest of the peptide. This pi bond will form a C double bond O, that group will leave, and we end up with the same thing again. CHR NH C double bond S and this becomes a C double bond O. And then we have the rest of the peptide. Now what's interesting, this molecule, it is referred to as a phenyl hydantoin. And phenylhydantoins have been identified with the different R residues based on the different amino acids. Now, that means we can isolate this molecule and identify it, and then proceed with the Edman degradation on the next end and identify it. And so we get the sequence from the N-Terminus, or the left-hand side of the peptide. The process actually has been automated so that you can do up to 50 units fairly quickly. Now, you say, well, why can't we just go on indefinitely? Well, the problem is, while on average the reaction takes so long, the hydrolysis will produce one phenylhydantoin, but a few times you'll get one or two. And so you start maybe hydrolyzing two of them. And so the major product, of course, is the one phenylhydantoin. But this error of some molecules reacting a little more than others creeps in each time more and more. It adds up. And so by the time you've done 50 units, you're not sure the next phenylhydantoin you've got, you're not sure which one actually is the next peptide, because you start cleaving off more molecules. The rate competitive hydrolysis of the other sections of peptide will eventually interfere with this process. So it's only good for about 50 units. Fortunately, we can also use a process starting from the C-Terminus. We can analyze from the C-Terminus residue. And if I do this, analyze from the C-Terminus or C-terminal amino acid residue. In order to do this, I use an enzyme called Carboxypeptidase. Now, carboxypeptidase is an enzyme that itself has 301 amino acid residues. Wow, big amino acid. So I'm not going to take the time to draw it here. I may show you a structure for this molecule in class, but I'm not going to here. 301 amino acid residues in carboxypeptidase. But this enzyme is designed to attach and attack to the C-terminal residue and catalyze C-terminal amino acid cleavage from the C-terminus only. So if I have a long peptide and I've got this amide unit, and I've got some R group here in water, and I hit it with carboxypeptidase, okay, I'll just call this my enzyme. Okay. What I will get, of course, is NH2, CHCO2H with its R group, plus the rest of the peptide. Now, we do have one problem with this. The enzyme immediately starts working on the other peptide. But if we use controlled conditions, we know the relative rate of reaction. We can actually analyze the solution, isolate it, and continue with reaction at every point. So the process can be automated. However, through careful experimental work, successful sequential analysis can only be achieved for the first 3 or 4 residues before partial hydrolysis, random variations in the rate, lead to lack of confidence in our determination of the sequence. All right, fine. So we can get 3 or 4 amino acid residues from the C-terminus. We can get up to 50 residues from the N-terminus. In the C-terminus, we use carboxypeptidase. In the N-terminus, we use the Edman degradation. But how do you determine the sequence for a polypeptide that has many more amino acid residues? Well, for larger polypeptides, what we do is we partially hydrolyze the larger peptide into smaller fragments. And then we determine the sequence of those fragments. Let's consider an example. We'll start with a simple one because it's just easier to work with. Suppose we had a pentapeptide. Okay. Now, suppose we didn't have the chemical capability of sequentially analyzing a pentapeptide. All we could do is analyze tripeptides. What we could do is we could take our unknown pentapeptide, boil it up for a short period of time, isolate all the tripeptides from it, and sequentially analyze them using a technique. And suppose the only tripeptides we found after carrying out that procedure was glutamine, phenylalanine, and tryptophan. In another one, there was aspartic acid, glutamine, and phenylalanine. And the next unique one was phenylalanine, aspartic acid, and glutamine. These 3 tripeptides were observed. Now, we can use these fragments to create a puzzle, if you like, in which we can assemble a pentapeptide. In other words, for example, I would write down my glutamine, phenylalanine, and tryptophan here. Okay. Then I would see this aspartic acid with glutamine phenylalanine could have come here. That is, there could have been a peptide that started with aspartic acid and you just broke it in different places when you did your partial hydrolysis. And lastly, we see the phenylalanine, aspartic acid, and glutamine lines up well with this. So when we were partially hydrolyzing our pentapeptide, different pentapeptides broke up in different places to yield the different tripeptides. Notice this has 1, 2, 3, 4, 5 amino acid residues, so it's a pentapeptide. Notice if we broke this one at the glutamine phenylalanine spot, we would have only gotten one tripeptide. But another pentapeptide could have broken in a different place. We can generate all these three tripeptides from a certain number of pentapeptide molecules. So by carrying out partial hydrolysis, sequentially analyzing for tripeptides, and then puzzle working to reassemble it, we've been able to identify our pentapeptide. Now there is also another technique which will allow us to, if you like, identify orders in these peptides. And that's utilizing specific enzymes which cleave amino acids in very specific ways. We can partially hydrolyze large peptides utilizing enzymes that cleave very specific amide linkages. Now, some are more specific than others. Okay? We're going to list these enzymes. I'm going to split what they cleave up into what you should know and what they are also known to do. Before the course, we're going to try and simplify it a little bit. The first enzyme is called Chymotrypsin. To understand the behavior of chymotrypsin, I'm going to draw down here a random peptide. I better raise it a little bit. We have the N-terminal with the NH. I'm going to move this over. I'm not going to have enough room. I don't want to draw the whole thing and run out of room. So N-terminals on this side. It is attached to an NH and an amide bond. CH, C double bond O. We're going to have a bond here. The off of the H will have an R. Now, this is attached to NH. We have a CH. I'm going to call this RR prime. C double bond O. And that's our C-terminus. We have an N-terminal, NHCH, C double bond O. Okay. Now, this position here, right here, is the bond we're going to cleave. The bond that we're going to cleave. Now, the question is, if we cleave here, note that we're cleaving to the left of R prime or to the right of the R. In other words, the amide bond that's being cleaved is the bond to the right of the amino acid that has the R group, or the bond to the left of the amino acid that has the R prime group. What does chymotrypsin cleave? Well, chymotrypsin cleaves to the right of phenylalanine, tyrosine, or tryptophan. So it cleaves to the right of these. Will only cleave those when the R prime group does not equal proline. Now, I'll be honest, chymotrypsin's also known to cleave when R is isoleucine, leucine, and valine, but we're not going to include those. We're going to keep it somewhat simple for this course. So, if the R group here makes this amino acid phenylalanine, tyrosine, tryptophan, it will cleave this bond to the right of it, notice, as long as the group with the R prime is not proline. Proline blocks the cleavage. Well, there's another amino acid called pepsin. Pepsin cleaves next door to the same amino acids, but it cleaves to the left of them. So, if R prime is phenylalanine, tyrosine, tryptophan, if R prime is phenylalanine, pepsin will cleave to the left of it, as long as R, the group with the R group here, is not proline. Proline seems to interfere with this process. Now, pepsin is even worse than chymotrypsin. It's very nonspecific. It's also known to cleave leucine, aspartic acid, and glutamic acid. There's another enzyme called trypsin. Trypsin, when the R group is lysine or arginine, trypsin will cleave to the right of it. So if you have a long peptide, find lysine or arginine and you can cleave to the right of those. Again, the other side of the bond cannot be proline. Now we've got two types of bromocyanogens. This is a laboratory reagent. We can use bromocyanogen in aqueous solution, or we can use bromocyanogen in anhydrous conditions. Bromocyanogen reacts with methionine. Cleaves when the R group is a methionine, cleaves to the right of it. Okay? There is no limitation with proline. In anhydrous conditions, it will cleave to the right of methionine or tryptophan. Note that trypsin, bromocyanogen, and anhydrous of bromocyanogen are very specific. They're very specific. Okay. And lastly, we have an enzyme called thermolysin. Thermolysin is an enzyme that will cleave when the enzyme R' groups are isoleucine, leucine, and valine. Note that the R group cannot be proline. The R group cannot be proline. Now, thermolysin is also known to cleave phenylalanine, tyrosine, and tryptophan, but we're just going to require you to learn these and apply these in this course. Now, so when you're using thermolysin, you find the R' group is isoleucine, you cleave to the left of it, as long as there's no proline there. So let's try applying this to a random, I'll just write down a random sequence of amino acids and ask what is the peptide you'd get. So let's just apply this really quickly. So I'm going to rip this out. And just remember, if the diagram at the bottom of the page confused you, just remember whenever you memorize the R', that means to the left of. And whenever you memorized R, that means to the right of. So, you know, let's say I've got alanine, and we've got tyrosine, and we've got proline, and we've got cysteine, and we got, I don't know, lysine, and we've got let's go another alanine and proline and arginine. Okay, and let's say we applied chymotrypsin to this. Where would chymotrypsin cleave? Well, R, I said, means to the right of. So find phenylalanine, tyrosine, or tryptophan. We've got a tyrosine, no phenylalanine, no tryptophan. So it would cleave to the right here. Except it says you can't have a proline for R'. That is, the bond you're cleaving can't be attached to a proline. So chymotrypsin would not cleave this molecule. What about pepsin? Pepsin would cleave to the left of phenylalanine, tyrosine, and tryptophan, so it would cleave here. Alanine's on the other side, so that cleavage would occur. So you'd get an alanine, and then you'd get this rest of the peptide. Okay. What about trypsin? Trypsin cleaves to the right of lysine and arginine as long as there's no proline. Well, to the right of arginine, I've got nothing here, so there's nothing cleave. To the right of lysine, I have an alanine, so I'd cleave here. So I'd get two peptides, an alanine, proline, arginine, which is a tripeptide, and I'd get a pentapeptide, alanine, tyrosine, proline, cysteine, lysine. So you begin to see how this is done. Okay. The last thing I want to mention in the amino acid section of notes is a consideration or review of what we covered in the thiol section last semester. And that is, thiols can be oxidized to produce disulfide bridges. Cysteine can react with another cysteine. And I draw it as an equilibrium. It's really not. We can oxidize, and we use O in square brackets to indicate oxidation, and produce a disulfide linkage. Now, last semester we talked about how such disulfide linkages were attributable, or the cause of, that to which we could attribute curly hair, or the lack thereof. If you had lots of disulfide bridges, this could hold curls in your hair. If you were on the thiol side, your hair would be straighter. Disulfide bridges are very important in proteins and peptides. Okay. When cysteine that are in close proximity to one another forms a disulfide bridges, you can actually form a loop in a peptide. For example, I'll draw this one. We have a cysteine, a tyrosine, isoleucine, a glutamic acid, an arginine, and another cysteine. And these two cysteines link up through a disulfide bridge. So those are the sulfurs from the cysteines. And off here we'd have a proline. Okay. A leucine. A glycine. And the glycine ends in your NH3+. Now, I admit I've drawn the N-Terminus to the right. You usually draw it the other way, flipped. But the point is, disulfide linkages can produce a loop. This peptide is called bovine oxytocin, a peptide involved in labor. In your notes, I have drawn the structure of bovine insulin. In the case of bovine insulin, you not only have one long peptide, which loops through, has a loop through a disulfide bridge, but you also have a very long other peptide in which there are cross-links between these chains. So cysteines can lead to cross-linking in peptide chains or loops. Bovine insulin is an example of a larger peptide that has both loops by disulfide bridges and loops and crosslinks, where two chains are held together. Okay. You're responsible for being able to recognize these two. You do not have to draw them. Obviously, I'm not going to make you draw bovine insulin. Well, that's it for amino acids. I'm Dr. Brian Lloyd. This is my Scribblecast. Thank you very much.